Hi, everyone. So today I'll be giving some research update on what we're doing at Ohio State University. So today I'll be talking about effect of a short day treatment on runnering and daughter plant production and mother plant vigor of Fronteras propagation transplants. So the overall goal of my project is to develop environmental strategies for year-round propagation in greenhouse, and we're working on objective 2.1. And specifically, our objective is to see the effect of artificial chilling in the propagation efficiency of mother plants. And our hypothesis is that artificial chilling of the strawberry propagation transplants will be economical to promote vegetative growth and runnering. So to test this objective of the effectiveness of artificial chilling, we conducted one experiment. We conducted an experiment to test the effect of chilling. So we had a control treatment in which I grew the rooted runner tips for 8 weeks, and then they were transplanted in the greenhouse, versus we had a chilling treatment in which they were established for 8 weeks in a long day and 26 degrees temperature, and then followed by a chilling period at 0 degrees centigrade and dark. And then we transplanted, and then to evaluate the runnering and mother plant after that. So we conducted this experiment for multiple years with Albion and Fronteras, Albion a long day type and Fronteras a short day type cultivar. So from this, we got consistent results for multiple trials in which we found that Albion, after chilling for 1,050 hours, had a significantly higher number of runners and daughter plants at 12 weeks after transplanting. So Albion, we chilled for 1,050 hours because of their knowledge from the conventional propagation system and how they chill the plants. And for Fronteras, we did 450 hours in the same way. And for Fronteras, we did not see any difference for runner, daughter plant production, or mother plant vigor, and the result was consistent for multiple years. So we already saw that the Albion showed positive effect, but Fronteras did not. And the Fronteras mother plant, this is what the plant looked like at 12 weeks post-runnering. So we had a plant that had a single crown and just 3 to 4 leaves, and that was throughout the runnering period. So a small plant canopy, and then had maybe 7, 8 runners, but again, chilling had no effect on it. So we wanted to work further on for this cultivar and to see why the plants did not sense the chilling, or why it did not show a positive effect. So looking at literature, what we found is that when the strawberry plants are in the field, they go into dormancy in autumn, late autumn, and then once the plant get a certain number of chilling hours, they're able to overcome that dormancy. And then there's a lot of hormonal changes, sugar changes in the sugar content in the crown roots, and then when the chilling requirement is met, and then favorable conditions are met in the spring, then the plants show a very high vegetative vigor, like they grow really fast because of all of those physiological changes. So we wanted to test the conditions to induce dormancy in strawberry transplant produced indoor. So we did another experiment. I'm going to talk specifically about Fronteras from now on. So we did, based on literature, we've found that the temperature to induce dormancy in strawberries should be at least 15 degrees centigrade or higher, and under short day conditions. So based on some of the European researchers and their work, we wanted to test 5 and 10 weeks of those dormancy inducing conditions. So the first dormancy, so the first treatment was just a pre-treatment for 5 weeks. So the pre-treatment was either a 9, a 16, or a 23 degrees constant temperature. for 10 weeks. And the second was the same temperatures, but for 10 weeks of, 5 and 10 weeks. And then we had a control, which was a no pre-treatment. So once the transplant was established for 8 weeks, they were directly transplanted. So 16 was kind of the threshold because of what we knew from literature. And we wanted to test a higher temperature, 23, because suppose this works, and then we're able to see that effect after pre-treatment and then chilling. We also want to have a method that's applicable in greenhouse, so that we don't need growers won't need a grow chamber to run it at 16 degrees. So which is why we tested multiple temperatures.
Which I have a question.
Yes.
The environment during the outside treatment, the white box.
Yeah.
What was like?
Like this one?
Yeah, exactly.
Oh,
for all of them.
this is just like nothing.
Where was the plan?
So because the timeline is different, so I would start rooting this plant first. So when the plants, and then this one at last. So there was not like, it was aligned in a way that I start at different point, but I transplanted at the same point. And because this was grown and the treatment was provided in a growth chamber, so we didn't have to worry about the environment conditions at that time.
When you quantify the size of the plants, were they similar or was it big?
Yes, there was, it was similar. I measured a number of leaves, crown diameters before transplanting and after the treatment. So yeah, we did not see difference in that.
Thank you.
Yeah. Yeah, so we did this experiment, and so what we found is that what we were expecting is that a dormant plant is usually has a shorter petioles and a smaller fresh mass as compared to a normal growing plant. And petiole length is the most typical trait of a dormant plant, having a short petiole length. But in our experiment, we did not see that difference of petiole length and fresh weight. I think it's because we grew them at a long day and high temperature. So over time, that dormancy is overcome by that condition. And then I also did starch and sugar analysis from the roots and crowns. And we found that after 5 and 10 weeks of treatment, the starch content and glucose content was significantly higher than control. And then another thing was we found that the number of crowns was significantly higher than control for both 5 and 10 weeks after treatment. This happened because during the transplant production and rendering, the short-day cultivar Fronteras was always in a long day condition and high temperature. So there was never a flower inducing condition for them. So if the flower is not induced and it's vegetative, then the crown branching won't occur, because for crown branching to occur, the terminal bud should be reproductive. So when there's terminal flower, apical dominance occurs, and then that's what shoots the side crowns. So because we had a short day treatment for them, so we were able to see that crown branching, and which we thought was interesting, and we wanted to plan another experiment based on this results. And also, even though we didn't see a typical morphological difference in the dormancy, we did see difference in sugar and glucose starch and glucose content. So we wanted to work on this further. So for the next experiment, what we did was we wanted to do the same short day treatment. So once we grew the transplants for 8 weeks in a long day, 26 degrees high temperature conditions, and the tray plant are about this size. We provided a short day and 23. We picked 23 to work further on this. And the thought was that we induce dormancy and then flower bud initiation. And then we have a plant that is a single crown to a flower bud initiated plant. And then we provide chilling after the plants are dormant, and then this will increase runnering, and then the mother plant will also have a multiple branch crowns. So this was the next experiment setup, and this, I will talk further about the treatments. So, yeah, control treatment, just a transplant established for 8 weeks. And then we also wanted to add a chilling treatment, which is the same as the first chilling experiment, transplants followed by 450 hours of chilling, and then a pre-treated one, which is first they were grown for 8 weeks, followed by a short day pre-treatment, 10-hour photo period at 23 degrees centigrade. And because we did see difference in 5 weeks, we did not think it was not necessary to use the 10 weeks, which is just an excess number of time for the treatment. So we just worked on 5 weeks. And then we wanted to see how this combination would work. So after the transplants were established, a short day treatment versus a chilling. So we had the combinations in a way that we can select what exactly is the factor that will affect the runnering of the plant. And then they were transplanted in long day conditions and high temperature for evaluation. Yeah, so 450 hours because we are working in Fronteras. So what we found from this experiment is that, okay, I'm gonna explain the graph. So the chilled one has the patterned one is a chilled, and then this is a control. And the solid color is a pre-treated, and the patterned is a pre-treated plus chilling. So the patterned graphs have a chilling factor to it. So we found out that the shorter treatment significantly increased runnering and daughter plant production in Fronteras. However, chilling did not have an effect, and then short day treatment was alone able to increase the runnering and daughter plants. So I think pre-treated increased by up to 37% for the number of runners produced. And talking about mother plant vigor, because crown branching occurred in Fronteras with the pre-treatments, both the chilling and without chilling, it significantly increased the crowns. We can see like higher than about 3.5 in average. And once the number of crowns increased, it also increased the number of leaves. So we did see a significant difference in that. And followed by, because we had the transplants that had a flower buds induced. So during the runnering period, evaluation period of 12 weeks, we did see flowers in the transplants with the short day pre-treated transplants for 3 weeks period. And yesterday we talked a lot about how this is a huge cost factor and growers don't want that. But as you can see, the number of flower trusses per plant is less than 1. And then it was only during this period of time. And then these were the flowers that were induced during the short day pre-treated. But then there was no other flower induction after that because we were growing them at 26 degrees centigrade. So after that, it did not affect or induce any flowers. So I think maybe it's not that bad, but I would want to hear from growers. And then another thing we measured is the leaf area of mother plants, and with the increase in the number of leaves, we also saw increase in leaf area of the mother plants significantly at 12 weeks after treatment. And yeah, we also saw a significant increase in the mother plant biomass with the short day pre-treatments. Again, no effect of chilling, and then we did not see a pattern in the runners. So this is what this is what the plants look like. The pre-treated ones had a higher, bigger canopy. And this is what they look like at the end of the transplant, at 12 weeks with the pre-treated ones having more canopy. So in summary, 5 weeks of short day treatment significantly increased runnering and chilling had no effect. And it did produce flower buds for a 3 weeks period during the runnering. So just to work as this is my final experiment. So based on our knowledge or our results from the long day type Albion and short day type Fronteras, we're currently working on this cultivar trial. And these are the cultivars from the core group that we're working on in the project, and also depend on availability. So for long day type, we just have two treatments, control and chilling, because we saw that effect of chilling was positive in Albion. And then for the short day type, we have a control, a chilling, and then a short day treatment based on our results from Fronteras. I think the long day type is pretty straightforward, but I will go a little bit about the short day type treatments. So for the control, again, this is the tray plan establishment. So for 8 weeks, the plans were established. And then for a short day treatment, in the previous experiment, we had 8 weeks plus 5 weeks of short day treatment, but we thought we don't want to have that extra time. So in order to do that, we kind of incorporated that 5 weeks in the transplant production. So the first 3 weeks would be a long day, 26, and then followed by a short day and 23 degrees temperature. And then because we saw the no effect of chilling in Fronteras, but we still wanted to test out the cultivars and maybe we'll see a difference. So we added a chilling treatments, and the plants will be transplanted in the greenhouse in October, will be done by December. So yeah, we are looking forward to this result, and I want to thank Dr. Kubota, all my lab members, Ibrahim, and NCSU team for sending us the plant materials. And that's all. Sorry, I was running out of time.
We have time for one question. Okay, no question. Pooja, well, good job.